So now we are going to see about ribosomes and cytoskeleton. So present in eukaryotic cell. So here ribosomes were first observed and discovered by George Pallade.
We have already discussed about George Pallade.
No. So he discovered these ribosomes. He observed these ribosomes and discovered in 1953. And so these ribosomes are composed of ribonucleic acid, that is RNA, and proteins. So these ribosomes, it lacks surrounding membrane. It does not have a membrane. It does not have a membrane surrounding it. Cell wall is just the surrounding plasma membrane in plants. So in eukaryotic cells, these ribosomes are of 80S, and prokaryotic cell it is 70S. So you have been seeing yes. So what is this yes? So it is nothing but yes, Swedberg's unit.
So Swedberg is a person or?
Yeah, Swedberg is a person, and he discovered this. So we have kept his name as Swedberg's unit. So it is nothing but sedimentation coefficient. That is, it measures density and size. So according to the density and size, it is given as for eukaryotic it is 80S, and for prokaryotic the ribosomes are of 70S.
Can you say 1S is equal to what?
1S is equal to 1 Swedberg unit.
No, that's right. 1S is equal to in centimeter or in millimeter or in micrometer. Can you?
So it is nothing but Swedberg's unit. So this he found this
units.
Units, and this is based on sedimentation coefficient. It is nothing but this Swedberg unit, 1 Swedberg unit, 1S is equal to 10 to the power minus 13 seconds.
So why they are relating time?
Yeah, it is because sedimentation coefficient.
Sedimentation.
How long it takes to sediment at the bottom. So how long the molecule, these ribosomes.
With that they measure the weight.
Yeah, with that they measure the density and size of the ribosomes. So this is about eukaryotes and prokaryotes. These are made up of 2 subunits. Like we saw in prokaryote is 50S plus 30S. So this is about ribosomes. And next we are going to see about cytoskeleton. These cytoskeleton are filamentous, proteinaceous and elongated extension structures present in cytoplasm. Cytoplasm of the cell. So here it is known as cytoskeleton. It gives mechanical support and motility to the cell. So what this cytoskeleton do is
it maintains the shape of the cell.
Yeah, it maintains the shape of the cell, it supports and it gives motility to the cell, etc. So this is the function of cytoskeleton. So next we discuss about Celia and Flagella in eukaryotic cells. So now we are going to discuss about Celia and Flagella which are present in eukaryotic cells. So here Celia and Flagella are hair-like outgrowth which are from cell membrane, which arises from cell membrane. So here Celia helps to move the organism or the surrounding fluid. It helps to move either the cell or the surrounding fluid. Where Flagella it helps only for the movement of the cell. It does not help to move the surrounding fluid. So in prokaryotes, prokaryotes also they have flagella, and eukaryotes also they have flagella. But prokaryote, the flagella which are present in prokaryotes are structurally different from eukaryotes. So now we are going to see the structure of only eukaryotic flagella, flagella and cilia. So here both flagella and cilia are covered by plasma membrane. So here they are covered by plasma membrane, and these both flagella and cilia both emerge from centriole-like structure called basal body in the plasma membrane. So here when you see the structure, for both Celia and Flagella, their core is called axoneme. Core is the center part of Flagella or Celia. So these possess a number of microtubules running parallelly along the axis. So if you cut a Flagella or Celia, we'll be getting a structure like this, which will be arranged parallelly like this. Okay, can you understand? So if this is a Flagella, if you cut like this is a part which you see. Okay. So here this is the plasma membrane which is covered, and they have a number of microtubules which are arranged in the 9+2 array. Okay. So what is 9+2? There are 2 microtubules at the center. Okay, so these 2 are connected by a bridge, central bridge. Okay. And also a sheath, central sheath encloses these 2 microtubules.
Like a bracket.
Yeah. Okay. And surrounding these, there are many peripheral microtubules. Okay, which are called as doublet because 2 microtubules are attached. So here these microtubules are 9 in number, so therefore it is called as 9+2 array. Okay, a 9+2 array. So here these peripheral microtubules, which is also known as doublets, are connected through inter-doublet bridge. Okay.
Double bridge like this. Okay.
Yeah. And here, so here these peripheral microtubules are connected to the central microtubule or the sheath through radial spoke. Okay, so this is the structure of both Celia and Flagella. Okay. So now in eukaryotic cell, in prokaryotic cell it is different. Okay, it has different structure. Okay. So now we'll discuss about centrosome and centrioles. So now we are going to discuss about the centrosome and centrioles present in eukaryotic cell. Okay. So here centrosome is an organelle. Okay. It is composed of 2 cylindrical structures called centrioles.
Okay.
So these centrioles are surrounded by amorphous pericentriolar materials.
So you will say about it in the next upcoming or?
Yeah, pericentriolar materials, they are connected by tubules as we saw in Celia and Flagella. They will be connected by pericentriolar tubules. There will be these tubules are like those tubules are 3 in number, triplet, and they will be connected with the central hub through radial spokes. Okay. So here these are surrounded by pericentriolar materials. Which one? Centrioles. Okay. So here both the centrioles, which are the 2 cylindrical centrioles, both the centrioles are perpendicular to each other.
So this is perpendicular. Okay,
perpendicular to each other. And it is made of 9 evenly spaced tubulin. Okay, tubulin or fibril. Okay. So the central part of this centriole is known as hub, which is proteinaceous.
So it has more protein content.
Yeah. So here these centrioles form the basal body of Celia and Flagella. We have already studied
the base.
It arises from the basal body. So yeah, that basal body is centriole. Okay. So it also forms a spindle apparatus during cell division. So during cell division, it forms a spindle apparatus, spindle kind of thing, which we will be studying in cell cycle and cell division. Spindle is a thread-like substance.
Thread.
Okay. So it produces spindle apparatus during cell division. Okay, we will be studying that in cell cycle and cell division. In the chapter we will be discussing about spindle apparatus and what its role in cell division and everything. Okay, so next we are going to the next subtopic, which is the nucleus. Okay. So now we are going to discuss about the nucleus which are present in eukaryotic cells. So here it was first discovered by Robert Brown. This nucleus was first discovered by him, and this material, the nucleus is stained by bacteria. Okay. So after staining, chromatin was found. Yes, inside the nucleus. So it was discovered by Fleming. Okay, chromatin is nothing but elaborate nucleoprotein fibers. So generally what are all present in these nucleus are chromatin, nuclear matrix, nucleoli, nucleolus. These are all present in the nucleus, okay?
Found by Fleming. Discovered by Fleming.
No, Fleming discovered only chromatin. Okay. So here nuclear envelope, okay, is of two parallel membrane. Okay. So the two parallel in between the two membrane, the space is known as perinuclear space.
The space is known as perinuclear space. Okay.
And this outer membrane, inner membrane, two parallel membrane. So this outer membrane forms, it is continuous with the endoplasmic reticulum.
Endoplasmic reticulum.
Yes, it attaches to the outer membrane, and therefore many ribosomes are present on it. Okay. So this nuclear envelope have some minute pores, have many minute pores at the sides. So where these two nuclear membranes fuses together and forms the pores.
Okay.
So here what does these pores do? These allow the passage of RNA and protein within inside the nucleus and outside the nucleus, both to and fro the nucleus. They pass to the passage of RNA and protein. Okay. So here cells with no nucleus. Can you name any few cells?
Cells with no nucleus.
No. We have already seen that there is RBCs of mammals.
Okay.
So red blood cells of mammals do not have nucleus.
Okay.
Of some mammals have nucleus, like if you see in amphibians like frogs. So their RBCs are nucleated. We have seen this already in structural organization animals. So generally RBCs of mammals do not have any nucleus, and also seed tube cells. This one also we have already discussed. Seed tube cells are cells which are present in all vascular plants, in angiosperms and in gymnosperms. So here what these seed tube cells do not have any nucleus. So what governs functions of these seed tube cells?
Okay.
It is companion cells. Okay. And this companion cells takes over it manages all the functions which are happening among the seed tube cells. Okay.
Okay.
So nucleoplasm, it is nothing but the nuclear matrix. It is also known as nucleoplasm. So this nucleoplasm contains nucleolus and chromatin and everything. So here nucleolus are present inside the nucleus. And these are non-membrane bound structures. These do not have membrane, and these are present in many number inside the nucleus. Okay. So what does these nucleolus do? These nucleolus
Produce active RNA.
Yeah, they are involved in the synthesis of active RNA. Okay. So they produce RNA.
They are part of producing RNA.
Yes. Okay, so these produces RNA, ribonucleic acid. No, not a part. This nucleolus mainly produces RNA. No other system produces RNA. Synthesis of RNA. And if there are more, nucleoli is also present inside nucleus. Okay, what does these nucleoli do? These nucleoli synthesizes proteins. Okay. So more the number of nucleoli, more protein synthesis will happen in a nucleus. Okay. So now we'll see what are the structures of chromosomes and the centromere. We'll be seeing that structures. So previously we saw about chromatin, right? Chromatin which are present in the stained conditions. So these chromatin have DNA and histone proteins in them. And in sometimes it has non-histone proteins and RNA also. Sometimes these chromatin contain. Okay. So mainly it contains DNA, histone proteins and non-histone proteins with RNA. Okay. So here human DNA, each cell human DNA is 2 meter long.
2 meter long.
Yeah.
So we can see it.
No. 2 meter long in a highly coiled manner. It is inside the nucleus. Nucleus itself we cannot, even a cell itself we cannot see. How can you see the?
Then how we can say it's 2 meter long?
Yeah, it is thread, fine thread, highly coiled inside the nucleus. Okay. fine thread, which itself you cannot see. It is highly coiled and it is present inside the nucleus. Okay.
Highly coiled,
Yeah.
so it's so small.
Yeah. Okay. So now, based on the position of centromere in a chromosome, it is of 4 types. Chromosomes are of 4 types. First one is metacentric chromosome.
Okay.
So here in metacentric chromosome, the centromere is present at the center of the chromosome. Okay. So this centromere, to the side of a centromere, a small structure called kinetochore is present.
Kinetochore.
Yes,
Okay.
which helps in cell division.
Okay.
So in metacentric chromosome, the centromere is present at the center of the chromosome like that.
Okay.
And in submetacentric chromosome, the centromere is present just above the center of the chromosome.
Okay.
And in acrocentric chromosome, the centromere is present near the side, near the end of the chromosome. It is known as acrocentric chromosome. And in telocentric chromosome, it is situated in the terminal position of the chromosome.
Topmost.
Yes.
Okay.
So it is known as telocentric chromosome. So with this, the nucleus gets over. Nucleus of eukaryotic cells gets over. And we are coming to the last subtopic, which is microbodies.
Okay.
So these microbodies are present both in plant cells as well as in animal cells.
Okay.
So these microbodies are membrane bound, minute vesicles present in both the cells.
Okay.
So what does these do? These contain various enzymes.
Okay.
That's all. These contain enzymes and these are small vesicles with membrane bound present in both plant and animal.
So what is the use of those enzymes? What
Yeah,
they
that
do?
enzymes they can digest.
Oh, they digest the
Yes.
food. Okay.
And they do many functions with that enzymes or whatever they do. So this is generally about eukaryotic cells. We have covered all the subtopics and all the organelles present in them, everything. So with this the chapter ends. Okay. So if you like this video, please subscribe and please click the bell icon which is nearby. If you have any doubts, please leave them in the comment section.
Thank you for watching.
Thank you.
Kritika and I will sign off.